This illustration shows a typical plant cell. As we already know, plant cells are eukaryotic cells. In this video, we are looking at the various organelles in eukaryotic cells. The cell wall. The cell wall is a rigid structure which surrounds the outer membrane of some cell. It is composed mainly of cellulose in plant cells. It forms a framework which protects, supports, and gives shape to the cell. The cell wall is permeable to gases, water, and other substances. Aside the plant cell, can you recall another or other types of cells that also have cell walls? You can review our videos to find out more. The cell membrane. It is thought that the cell surface membrane is composed of a bimolecular lipid fat layer interspersed with globular protein molecules. It is not rigid. The fluid mosaic model shows its structure. It is flexible and encloses the cell. It also controls the movement of materials into and out of the cell by way of its partially or semi-permeable membrane. This illustration shows the fluid mosaic model of the cell membrane. The lipid are the two layers as seen, the outer surface and the inner surface, and the globular protein molecules are found in between them. There are also larger pores, as you can see in the illustration. The cytoplasm consists of a watery ground substance containing various organelles and cell inclusions. The ground substance is clear and it is 90% water with salt and sugar dissolved in it. It contains fats, proteins, and enzymes. Cytoplasm is not static, that means it moves about. When observing living cytoplasm, organelles appear to move about due to cytoplasmic streaming, that is the mass movement of the cytoplasm. The nucleus is the control center of the cell. It contains hereditary material called deoxyribonucleic acid, DNA, which determines the characteristics of the cell. There is also another nucleic acid called messenger ribonucleic acid, mRNA for short, in the nucleus, which carries instruction from the DNA into the cytoplasm during protein synthesis. Nucleolus. The nucleolus is a spherical body in the nucleus which contains ribosomal RNA, chromatin, and protein. This is where ribosomes are produced. Nuclear envelope. The nuclear envelope is a double membrane structure which contains pores. It surrounds the nucleus and separates it from the cytoplasm. Mitochondria. Mitochondria are minute spherical or sausage-shaped organelles. Each mitochondrion is bounded by two membranes separated by a fluid-filled space. The inner of the two membranes is folded into cristae. The cristae provide a large internal surface area. On the surface area of the cristae are enzymes, which are involved in the release of energy in respiration. For this reason, the mitochondria are sometimes called the powerhouse of the cell. This illustration shows how the mitochondria looks like in the electron microscope. The number and location of mitochondria in the cell depends on the cell's energy requirements. Cells that produce a lot of energy contain many mitochondria concentrated at the site of energy use. Example, near the base of the tail of a sperm. However, most cells contain hundreds of mitochondria. Lysosomes. Lysosomes are very small, spherical, fluid-filled bodies bounded by a single membrane. They are present in the cytoplasm of most eukaryotic cells, but are particularly abundant in animal cells. They contain many enzymes involved in intracellular digestive breakdown during growth and repair of cells. These enzymes are also involved in the cellular defense against attack by bacteria, viruses, and toxic substances. Lysosomes are involved in autophagy, that is the destruction of parts of a cell. Example, worn-out organelles, or autolysis, that is the self-destruction of a cell. Endoplasmic reticulum. The endoplasmic reticulum is an elaborate network of membrane-bounded channels running throughout the cytoplasm. It is continuous with the nuclear membrane. There are two types of endoplasmic reticulum: rough or granular endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum has a rough surface due to the presence of ribosomes. The surface of the smooth endoplasmic reticulum has no ribosomes. In short, rough endoplasmic reticulums are called rough because they have ribosomes on their surface. The smooth endoplasmic reticulum is not associated with ribosomes, although most cells may contain both the rough and the smooth endoplasmic reticulum. Their numbers and relative proportions vary. The rough endoplasmic reticulum is more common in cells that secrete enzymes. Example, the pancreas and the salivary glands in mammals. If you recall, they secrete the pancreatic and the salivary amylase respectively. The rough endoplasmic reticulum functions as a supported platform for the ribosomes and as a transport system in the cell. Reading from the second paragraph, the rough Endoplasmic reticulum functions as a supporting platform for the ribosomes and as a transport system for the protein produced by them. The membranes of the endoplasmic reticulum serves as conducting pathways for the movement of other materials in the cell. Ribosomes. These minute organelles are found in large numbers in the cytoplasm of all prokaryotic and eukaryotic cells. Some occur freely in the cytoplasm, while others are bound to endoplasmic reticulum as already described. They are composed of RNA, ribonucleic acid, and protein, and are the sites of protein synthesis in the cell. The ribosomes bind amino acids together, forming polypeptide chain. Golgi body, or also known as Golgi apparatus. Each Golgi body is a stack of flattened membrane-bound sacs, cristae, singular cristae, with a cluster of vesicles at the edges. They are present in the cytoplasm of all eukaryotic cells, but absent in those of bacteria, that is, the prokaryotes. Golgi bodies produce cell membranes. Dictyosomes. The dictyosome is only found in plant cells. It is analogous to the Golgi bodies in animal cells. This means it performs the same role as the Golgi body performs in animal cells. It is made up of a stack of vacuoles with rounded vesicles on the sides. During cell division, the number of dictyosomes increases to produce vesicles whose polysaccharides are needed for the formation of cell plates. This plate develops into the plant cell wall. Chloroplast. Chloroplasts are large egg or disc-shaped double membrane-bound structures found in the cytoplasm of some plant cells. They contain the green pigment chlorophyll, which captures the light energy used in photosynthesis. A chloroplast has the following parts: external double membrane, Liquid stroma, grana, singular, granum. Integral thylakoids, starch grains. The stroma is the matrix in which the grana are suspended. It is the site of the dark stage of photosynthesis. The grana contains the green pigment chlorophyll. They are made up of disc-like membrane called thylakoids. The thylakoids pass through the stroma. An integral thylakoid linking neighboring grana. The light stage of photosynthesis occurs in the thylakoids. Vacuoles. Vacuoles are fluid-filled sacs bounded by a single membrane. They are found in all cells, but they vary greatly in size and function. Animal cells contain relatively small vacuoles in comparison with those of plant cells. Their functions vary. For example, food vacuoles, autophagic vacuoles, and contractile vacuoles. The vacuoles of the plant cell are large and are surrounded by a membrane called tonoplast. They contain the plant sap, or simply called sap. Centrioles. Most animal cells and some protozoes, example the algae, contain small rod-like structures which lie in the pairs in the cytoplasm near the nuclear membrane. These are the centrioles. A centriole is made up of 9 triplets of microtubules which are arranged in a ring forming a hollow cylinder. Each pair of centrioles lies close together with their axes at right angle to each other. Centrioles separate and move to opposite poles of the nucleus at the onset of cell division. They are believed to be responsible for the formation of spindle fibers, also composed of microtubules in animal cells. Cell inclusions. The non-living chemical substances found in the cell are referred to as cell inclusions. They are not membrane bound, so are not part of the membrane system. They exist in soluble forms in the vacuoles and as insoluble substances in the cytoplasm of both plant and animal cells, and in the cell wall of plant cells. Examples include carbohydrates, proteins, vitamins, fats and oils, food materials, tannins. Carbohydrates occur as sugars in the soluble form and as starch in insoluble forms. Starch forms minute grains of various shapes in the cytoplasm of plant cells. Proteins occur both in colloidal solution and solid form in cells. Plant cells store solid protein and enzymes in aleurone grains. Fats and oil occur as globules in cytoplasm. Tannins are found in solution in special cells and plant cell walls, and are also present in large quantities in dead plant tissues. Resins are yellow